discuss about the topic Qtl Fine Mapping. What is Qtl Fine Mapping? Means identification of the QTLs more precisely due to more recombination and identification of very closely linked markers. This procedure is called as Qtl Fine Mapping. For example, you take from this region to this region, you are getting one large QTL. It is think that this is nearly like a 20 centimorgan distance. But your gene is present in this one, in this region. Your gene which is responsible for your trait, in this case actually tannin, the gene which goes for the tannin concentration in sorghum, is in this region only. But how to, but you are having marker 1 here and marker 2 is here. But how to identify the marker which very much nearer to this particular gene? It is called as fine mapping. Fine mapping means we are going more precise. Fine means finest, like we are like finest word we will use in. It's like it's more precise. Like see, this CT105 and CT107 are the marker which are tightly linked with this particular tannin gene. But this is identified by the procedure called as Qtl fine mapping. I hope everyone understood what is Qtl fine mapping.
Sir, one doubt.
Just yeah.
Sir, we get a LOD graph, no, sir?
Yeah.
A continuous graph which has some threshold level. Can we just say the maximum threshold level we get will be the exact position of the marker, sir? It is not possible, sir.
But your marker 1 is here, marker 2 is here. From this region to this region, you are getting this one. Actually, from marker 1 to marker 2 LOD. It is not like this. Here it is going highest peak. This region is not, you can't say like this. Okay. But ultimate objective is what? You have to use this marker in the plant breeding, MAS. That's what I'm repeating every class. Okay, you should follow. See, this marker 1 is here and marker 2 is here. You have to use this marker directly in marker restricted selection. But this is this much distance from this particular gene. There may be chance of recombination. You may selection will go wrong. For higher reliability of the selection, we need a tightly linked marker. Okay? Always think from simplified way. Okay. So don't think always in a complex way. Okay. First, you understand the surface level. What is there actually? Okay. Directly, you don't dive into the very deep things. How it will be. How don't make like the in that detail, like in that deep sense. If you understood, it will become very complex. You are not going to get anything. You have to think from like surface way. First, you think. Here, your marker 1 is there, and your marker 2 is there. But your gene is here somewhere. You don't know. But you should go very nearer to them. For example, these markers, if you go identify very nearer marker, it is very easy to do selection. Like by using this marker, your reliability of the selection will be very high. That's why you have to go for fine mapping. It's not like that here you're getting to highest load, maybe okay, you're getting position. Then what you will do with that position? What? Okay.
Yes,
Ultimately,
sir.
identifying the marker nearer to the G is very important. That one point you should remember always. Whatever the things we are doing, all the purpose is same. Ultimately, we reach to a single point only. Identifying the marker within the gene or marker very near to the gene. Its ultimate aim in molecular breeding. If you identify, if you validate the gene and you identify the marker within the gene, it's super actually. It's very good. But it is the only the different difficult task. Now understood what is Qtl fine mapping? Just
Yes,
going
sir.
very much nearer to the gene of interest. Okay? You are identifying a very closely linked marker. Just remember this. You are identifying very closely linked markers for our gene of interest. This much you remember. And it is published in this, actually this work is published in PNAS. It's a very, very reputed journal actually. PNAS.
Sir, why it is difficult to find a functional marker, sir?
Actually, because it's very difficult. See, we are doing analysis. Um, the marker which are present within a gene, you may get, but it is very difficult. See, for example, if you do, because we are using a mapping population, max to max we are using like 200, 300, 400. The recombination will be very less. If you use larger population, larger marker, you may get functional marker also. That is not the problem. But normally in every map, uh, every work we will do, we are using a limited population and limited number of markers set. That's why identifying the probability of identifying the functional marker itself is very, very low. And if you are using SSR marker and they are most probably there in the non-coding regions because they are repetitive genomes. SSR means it's a repetitive genome. They are maybe present in the non-coding regions, but almost genes are coding one. They are present in the exons. But your all markers they are in the intron or a repetitive genome. They are not in the coding region. That may be the one case. And the population we are going for this one, it is like population. Sorry, population we are using here. It's very limited number of recombination. The resolution of maps will be very less. That's why identifying the marker within a gene are it's very, very difficult. Understood? That's why we are going for association mapping. This is the one of the limitation in the linkage mapping. Because we are doing controlled crosses, there will be a limited recombination. That is a disadvantage actually. That's why we are going for association mapping. We are going for some more advanced methods combining different next gen techniques like next generation sequencing plus mapping. All we are doing because of this one reason to get more link identify functional marker or more tightly linked marker. It's a good doubt actually. Okay, now you understood?
Yes, sir.
Then how to do it? I told like we are identifying a very, very tightly linked marker. As he asked, it's very difficult actually. We need to do a lot of circus to identify the marker within a gene are very nearer to gene. Homozygous lines. First, we have there is be a 6 different methods are there. Not only these 6, there are several other methods coming up every day. But in AKSing and BDSing, these are given just because exam purpose they are very important. I am discussing. First method is creating the homozygous line derived from near isogenic lines. You develop nils, contrasting nils, nil 1 and nil 2. Then you cross this nil and you will generate an F2 population. Then you do mapping in this population because when you are crossing the nils, they are differing only with the gene of interest. Further, you are recombining. If you are crossing nils, means there will be chance of recombination within this gene is very high. If you do mapping in that recombination population, the probability of identifying marker with this gene is very high. That's why we are using homozygous lines which are derived from crossing two nils. Then another one is intercross rails because rail population itself is a very having the more recombination like more than two recombinations created by more than two rounds of recombination. Further, you are crossing two you are crossing, you are intercrossing the real population, means it will go for further recombination. Means further resolution will be going to increase. Means you are going very nearer to the gene is very high. The chance is very high in case of intercross rates. Then we can use a genetically heterogeneous stock. Actually, it's intercrossing of the individuals and they maintained in the random mating. Then another approach is multi-parent advanced magic population. I did in detail. I have discussed what is magic. It means there will be eight founder lines. You cross among them, then you cross among these lines. Then you cross among this line. You develop one population. Means eight different parents will recombine and with sixth generation, and you are developing some population. Means higher will be the recombination. Ultimate only one point you should remember. Higher will be the recombination, more will be the resolution, more closer you will going to gene. This is one point is for common for every step, every different method. Okay, you are developing magic because to increase the recombination. Then reverse Qtl mapping. Actually, this is very important. Means it's a two step screening of a very large population like 2000 segregating population. Means why you are going for very large population? I told so many number of times. The reason for growing large number of population, I think
High
nearly 10
resolution.
times I have. Yeah,
High
more
resolution.
recombination, higher resolution. Means the individual with, you can identify the individual with all recombinants. For example, there will be, you take, if one gene is there, one segregating gene will be there. How many number of genotypes you are going to get? Or how many number of phenotypes you are going to get? How many number of phenotypes you are going to get? 2 power n.
Hmm.
And the genotypes you are going to get is 3 power n.
3 power.
How many number of population you are going to get? 4 power n.
4 power n.
If one gene is there, you are getting 4 power 4 population. If two gene is there, you are getting nearly 16 individuals. If three gene is there, 64 individuals. See, normally, if the two individuals are you are crossing different individual, at least see in rice, number of coding genes is nearly 40,000 genes. 4 power 40,000 genes means how much genes it will going to segregate. How many number of population you need for getting each recombination. That's why more will be the population size, more will be the probability of identifying. Tapping all the recombinants is very easy if you're having more large population. Then, but if you want to screen all 2000 population with, for example, you having 1000 markers, it's very difficult. I told why it is difficult. That's why what we will do, out of 2000, we sample out 200 individuals, like random sampling. Like there will be a lot of population. We randomly sampling 200 individuals from 2000. Then we do QTL mapping in this 200 individuals with a large number of markers, like 1000 markers. Then you are getting one major QTLs. Then after getting major QTL, you will get a flanking markers. Like this, you are getting one flanking markers here. You screen these flanking markers in the entire 2000. Wait, I had to. Okay, sorry. Where I was? Okay. Uh, okay. Actually, after identifying the flanked marker by using a 200 population with 1000 number of marker, just for example, I'm saying, but small sample population, large number of marker, you are identifying a flanking marker for a major QTL. After use this marker and you run this marker in the 2000 individuals, 2000 individuals, then you are going to get a genotypic data. Then you identify QTL in that. What are the QTL you are going to get in that that one? They are maybe your high resolution QTL, means very nearer to the gene of interest. This is called as reverse QTL mapping. Okay, understood how to do this reverse QTL mapping?
Yes, sir.
Then you can use combination of QTL mapping with transcriptome profiling. Transcriptome profiling means, what is transcriptome?
The sum total of mRNA produced in a cell.
Only mRNA,
Whole RNA.
whole RNA. That's right. Okay. Sum total of RNA present in the cell. RNA present in the cell. It's, if you're understanding, you're studying what are the RNA present is, how much level of expression of the particular gene by analyzing the transcriptome of the particular cell or a particular individual at particular stage. Means you having an expression data here, which gene is highly expressing, which gene is lower expression, at what position, what gene it is highly expressing, we are going to get. And we have a QTL map also. Like this one at one region, we have QTL map. Check within this region, which gene is highly expressing, which gene is highly downregulating, which gene is highly upregulating. The gene which are highly upregulating or gene which are the regulation of the particular gene is very different, and we have getting maximum upregulation or maximum downregulation in this particular region. That gene may be your gene of interest, maybe candidate gene. By this way, out of, for example, in this region you having 20 genes. For example, in this particular region we having 20 genes. By comparing with transcriptome data, in transcriptome data we are getting that the gene expression profile. Gene expression profile. Means which gene is highly upregulated, which gene is downregulated, we are going to get. And in out of 20 genes, like 2 to 4 genes are showing a different regulation. But all the genes which remain in baseline, no upregulation or no downregulation. Means that you can see that okay, these 2 and 4 genes in this particular region out of 20 genes may be your gene of interest. Then you just do sequencing. Then you identify some SNPs. You design caps marker for them and use it in a plant breeding. This is how you can combine QTL mapping with transcription profiling. Understood how to use transcriptome profiling in QTL mapping to fine map the candidate genes or fine map the candidate map? Understood?
Yes, sir.
Can you list out what are the different 6 methods?
First one is
Will
Niels,
F2
sir.
population.
Yeah, we need to generate magic population, or we need to generate
Niels.
homozygous derived nil derived from crossing nils. Then we can use intercross ril population. Then we can use magic
Heterogenetic stock.
heterogenetic stocks. We can do reverse QTL mapping.
And last
Then
is transcriptome profile. Combining QTL mapping.
transfer. Either you can give QTL seq, either RNA-seq, map. These things are there. I have this. I will have this map, map gap. There are so many things. Shore map, BSR-seq, selective genotyping. There are so many things are there nowadays. Okay, but you just remember what are there in the book. This is about Qtl fine mapping. What are the disadvantages of Qtl mapping? We discuss advantages, what is the advantages, how to use in the plant breeding, discussed in detail. Applications and advantages. What is the limitations? Lower is, I think we discussed, no? This is a very low, see, in every Qtl mapping, where even though you fine map, how much resource we need, how much effort we need, you're getting very large region. Because we are using a biparental mapping population or multi-parent. Means there will be a limited recombination. The resolution will be very, very low. This is the one of the major disadvantages. Resolution is very low in Qtl mapping. This is one of the biggest disadvantage of the Qtl mapping. And developing a mapping population. For each trait, you have to develop a mapping population. You want to do nematode resistance, you have to do mapping population, you have to create one mapping population. You have to do blast, you have to create another mapping population. You have to identify genes for blight, you have to create another mapping population. For every this one, you have to create different mapping population, then identify marker, then pyramid into different single line. Then you develop one variety which are resistant to nematode, blight, and blast. See this much, how many years it will take? You have to validate it. It's a very, very lengthy process. This is one of the disadvantages. It's a laborious, time consuming, costly. We have to develop a biparental mapping population for everything. See, for example, you take a mango tree. My senior is working on mango. My friend is also working on mango. If you want to develop a real population in mango, how much difficult it is? Can you develop real population in mango tree?
Very
No,
long
sir.
time it will take.
Very
So very perennial. So perennial, you may 2 terms will go for a scientist maybe. Have to burn 2 times to develop real population. That's why it's a perennial some the crops which are perennial in nature. We can't able to develop a QTL maps. Sorry, a real population or a biparental mapping population. It's very difficult. In that case, how to do QTL mapping? There are several QTLs for diseases in mango, or people will do QTL mapping in mango also. What are the approaches? There are different mapping populations for this, like pseudobacros, pseudo F2. When I will dealing with the tree breeding, I will deal with this. No, I will not going to deal. But pseudo F2 means because mango maybe the almost all tree crops are mango crops are heterogeneous. If we just use them directly, like if you F1 may be directly used for mapping sometimes because they are pseudo F2s. Even though they are F1, they are like highly segregating in nature. Where it is. Right, I had to take another. Sorry, actually had to. Okay, where I was. This is one of the resolution is very so one of the disadvantage of the QTL mapping. There is another method called as AB QTL approaches, advanced backcross QTL analysis. It is actually discovered by Tanksclay and Nelson in 1996 itself. It's just a combining a QTL mapping with breeding. Then it uses the backcross mapping population and development of promising lines for QTL. Like you can directly use QTL analysis to the breeding. How we will see. For example, you are creating parent 1 and parent 2, then you are generating F1, then you will generate a BC1 population. Then you backcross to any one of the parent, you continuously generate BC2, BC3. In BC1 and BC2, you do QTL map and you are identifying a QTL and the marker associated with your trait. Then you screen this marker after that next BC3 like this. Then you can, in process of developing a MABB scheme, marker arrested backcross breeding scheme only, you are developing a QTL and you are using within that population. This approach will consume like it will conserve or a limit, save the time of developing real population, identifying the QTL, then validation utilized in the breeding population. It will save your time because from mapping population to breeding population, first you have to identify marker in mapping population, then you have to use in breeding population in conventional QTL mapping. But in AB QTL mapping, you directly generate the breeding population BC1 and BC2. Here only you identify the QTL and here only we are using the next coming population so that your time will be saved. This is the one of the method called as advanced backcross QTL analysis. Okay, this is one method I want to discuss today. I hope everyone understood Qtl fine mapping, AB Qtl analysis. Any doubts? No one is there.
Sir, do we have to study this BSR sec, RNA sec?
Yeah, no. Actually, for just knowledge purpose, I'm telling. Maybe question will arise, but don't go in detail about multi-breeding. What I'm teaching in the slides, that much is enough. And read AK Singh and BD Singh book. What is there in that book, that much is more than enough. Okay. In PhD, if you are entering into the PhD, there will be one separate course called as genomics in plant breeding. There you are reading in this much detail. Okay. For XRFI, it is not needed to read all those things. QTL-seq, understand. But you have to know QTL-seq. QTL-seq is mostly used approach, and everyone in MSc only they are doing QTL-seq in many labs. At least you know what is QTL-seq and what is the advantage over the normal conventional QTL mapping. That much is more than enough. And watch my video because I already made a video on QTL-seq. For exam purpose only I made. That much is more than enough. QTL-seq and BSR-seq. Watch that video. That much is more than enough.
Okay, sir.
Actually, today it's very short class. I had to leave for somewhere. Friends are calling. Any doubts up to here? This much only for the QTL. If you remember this much, what is QTL definition? Who coined the term? Types of QTL mapping and QTL fine mapping. What are the different approaches of QTL mapping? AB QTL analysis, QTL seq. I will discuss. Watch my video and basic advantage and disadvantage of every method. It is more than enough for examination purpose. For ASRP, it is don't read this also. Okay, you just remember what is the definition of the QTL, scientist involved in it, and which are the mapping population used for QTL mapping. More than enough for ASR. Even don't read single marker analysis, all those advanced ABQTL, single marker analysis, composite neutral mapping. That is not needed for ASRP. For SRF, you have to read in detail. Okay. I'm closing the class today. You ask any doubt, we have 5 minute time. Tomorrow I will discuss association mapping. I will actually Monday, tomorrow maybe I will not take class. Association mapping, my friend will take. I ask him to take. But even I also, if we not free, I will take because I already have slides. After association mapping, we directly jump into the MAS schemes. Then after that, maybe I will teach uh, recombinant DNA technology. I will try to
Uh,
finish. I think within
Gene
15 days, I will
cloning,
try to finish all unit 5. Okay.
gene cloning will also come under RBNP concept.
Yeah. Recombinant DNA technology only it will come. In that only I will deal all those things. Biotechnology. Like I think I have to discuss that. See what is NGS. All those things I will discuss in biotechnology. It's a kind of repetition thing. That's why I avoided. I think I have another 2 minutes, 3 minutes time. I will discuss some questions related to Qtl mapping. Yeah. This one you answered. Behavior of chromosome during mitosis can account for Mendel's laws of segregation and independent assortment, and homologous chromosomes separated and allele segregate during meiosis. Linkage and
Option B,
crossing,
sir.
yeah, because in mitosis it will not happen. Segregation and independent assortment. It will happen in meiosis. And homologous chromosomes separate and alleles during meiosis. Only B is correct. Then, which of the QTL mapping methodology uses see exactly copy paste?
Reverse
Reverse
QTL
skipping.
mapping.
Yes, sir,
Yeah,
exactly the thing.
yeah, actually. Then the genetic mapping gives a relative position on different gene based on recombination rate, and mapping of recombination mapping always gives details about actual chromosome position of the genes. I discussed so many times. You had to tell them.
Statement one is true, sir.
Yeah,
Statement two is not false.
yeah, because it's a physical map. Actual chromosomal position is physical map. It's not linkage map. Then I think this is actually I have not covered. This is also not covered. This is also not covered. Okay. This is also not covered. Hmm. Which of the population used for mapping populations?
B, C, and D, sir.
Yeah,
BCND.
because F1 normally will not use F1. Okay. All other things are B, C, D will use. A only will not used. Plus F2 will use, F2 derived F3 will use, near SHN clients also we are use. But F1 will not use because F1 we are not getting any segregation. Then requirement of marker arrested backcross breeding. I discussed MAS, similar points. Higher
Sir,
heritability,
B
right?
and C definitely come, sir, and A, I don't know, sir.
What?
But options B and C are correct, sir.
How?
Not this question, sir. The next one, sir. Back cross.
Oh, you told, I was discussing. I confused whether you were telling about. Okay. The higher heritability, tightly linked marker with target gene, flanking markers, multi-location trials. What is the major requirement?
Heritability
A,
means
No. Choose the correct. So many options. Three options. Three are
sir,
correct.
fourth
Out of
option.
four.
Fourth option, sir. A, B, C.
Yeah. Because if you're using markers, no need of going for multi-location trials. Okay, because we are doing indirect selection. Marker genotype is similar in all the environments. Okay. When genes for bacterial blight resistance, so very
XC13,
simple.
XC21.
Yeah. And source also you should remember. IRBB,
So
IRBB source. Okay. Then the main effect QTL explains more than 10% of phenotypic variance. And QTL affecting RNA transcripts are called as expression QTLs. Both statements
Both
are
are
correct. I think I have discussed. discussed in our last class. In Qtl mapping, appropriate statistical, we are doing both genotyping and phenotyping, and we need statistical packages for both of them. Then we discussed it is actually multiple interval mapping because in this only we are able to identify epistaticus. The all rest other three methods we are not using. But actually answer keys was given wrong. Maybe he confuses like here inclusive composite interval mapping if there, then two option will be correct. But here they given only composite interval mapping. Even in original papers also you check, there is no, we can't do epistaticus by these methods. Only epistatic QTLs identified in multiple interval mapping or inclusive composite interval mapping or any multi-trait or multi-pulse QTL mapping only. Then this is again association mapping. This is association mapping. This is genomic selection. From molecular breeding, this much question only from 2021 to 2023. I think I may have missed some marker questions, but this much only. And see how much this much easy. Don't go very detailed. Okay, what I am discussing and what is there in the casing and BDCing is more than enough. And this time they have asked, I not kept. I think I have kept. Watch my videos. I think another two videos here also I'm making on SRF 2024. There is a from markers. So entire molecular breeding. They asked only two questions this time. One is on LD and sorry, three questions. One is on genomic selection, one is on MAS. No questions on QTL mapping. They asked on reliability of the selection. They given R percent value of the marker. 1 minus 2R is the formula. If you put that, you'll get some answer. I will discuss after discussing this. So this much is for today's class. I have to go there. Okay, bye. Bye everyone. Good night.
Good night,
Right,
sir.
sir.